Okay, thank you, Tanya and EJ. So I'm Eric Enslyn, professor of chemistry, specifically an organic chemist. And I'm going to tell you a little bit about what we've titled differential sensing. This is a technique that kind of marries supramolecular chemistry with what I call chemometrics. Now, chemometrics has become kind of synonymous with machine learning lately. Machine learning sounds a lot more attractive than chemometrics, but it's taking cross-reactive arrays of sensors and developing patterns that become diagnostic for a particular analyte or diagnostic for the consistency of a complex mixture. And the inspiration really is just the mammalian senses of taste and smell. To give you a little insight, our sense of taste really only has over here, maybe 5 different sensations: sweet, sour, salty, bitter, umami, or savory, delicious taste. And we mix and match the extent of response of those sensations to register a flavor. And there's not a highly selective receptor sitting on the tip of your tongue waiting for any particular sugar. It's that it's a composite response of many cross-reactive receptors that become diagnostic for a sugar. And our sense of smell, maybe 1,000 sensations or so, but none of those sensations are targeted to a selective specific molecule. So instead, you get a pattern of responses that we then register as an odor. Well, we would like to do this for a variety of different kinds of solutions. I'm going to say that what my group is probably most well known for is wine. We've developed a whole lot of diagnostic systems for wine that tell you about the varietal of the wine, even the hang time of the wine. But where that might be interested in wine for wineries, for medical health or something that I imagine this group might be more interested in. We have also targeted what are called the mitogen activated protein kinases. This is a project that's in collaboration with Kevin Dalby in our pharmacy department here at UT, where Kevin is able to express numerous of these MAP kinase proteins, and he works hard to understand what kinds of inhibitors and drugs can both activate or turn off a particular kinase. Because kinases often go awry in their expression in a variety of different cancers and these other kinds of human diseases listed down here. But now, what does a kinase do? It basically does a phosphorylation of either a serine, tyrosine, or threonine. So there's serine kinases, threonine kinases, etc. And the phosphorylation becomes part of a signaling, cell signaling. Well, many years ago, the Barbara Imperiali group developed a sensor that basically turns on when a phosphorylation takes place on an amino acid by what's known as a chelation-enhanced fluorescence increase. Phosphorylation occurs and magnesium binds this ligand and fluorescence turns on. So we're going to use that, in essence, to do this kind of patterning that I've alluded to. But we're going to pattern now kinase activity. And I've already told you that what our group is probably most well known for is wine, but we've also patterned various kinds of terpenes in perfumes. We've patterned various kinds of tannins that are in teas. We've patterned glycerides that are in adipocyte extracts. And the patterns are these kinds of plots here that are known as score plots. What we've done is take an optical response from an array of receptors. The receptors are arrayed in a well plate None of these particular receptors are selective for any one analyte. Instead, as implied by this cartoon, they're all cross-reactive. And when they cross-react, they give an optical pattern where that pattern is interpreted by chemometrics in the form of score plots. These are principal component or linear discriminant score plots that allow us to classify either what lipids are in adipocyte extracts, what tannins are in teas, etc. I'm going to employ that same technique to kinase activity. So what we've done here is take a variety of peptides where these peptides bind to different kinases, such as the ERK family of kinases, the J and K, or P38, as I'll show you in a moment. Those are families of kinases. But these peptides bind to all those families of kinases, yet they bind to them all differently. That's the origin of differential sensing. So when these peptides, 5 of them, bind to upwards of a dozen different kinases, the fluorescence starts to turn on using that chelation enhanced fluorescence strategy I showed a couple of slides ago. And we could start to generate patterns of the fluorescence turn on that become indicative to what kinase is active. So here now I'm showing some score plots of the variety of kinases that Kevin Dalby has supplied to us. When using those 5 different peptides, I want to stress that every single one of those peptides is actually phosphorylated by every single kinase. It's simply they're phosphorylated to different extents. And you need machine learning to extract the patterns that will identify the activity of the kinase and the identity of the kinase. I'll start wrapping up in a couple of minutes here, but we can not only identify the activity of the kinase, that's what we're showing here is an actual activity plot of the kinases. We can also plot inhibition of a variety of known kinase inhibitors, such as people develop for cancer therapeutics. So we're adding a known inhibitor here and also getting an activity plot of the inhibitor concentration versus the actual predicted inhibition of a individual kinase. I'll end by saying this is not done just simply in vivo, ex vivo or in vitro. We also are able to explore actual cell lysates and tumor extracts from animals, once again in collaboration with the Kevin Dalby group, who grows the mice and has the tumor extracts and puts the tumor extracts into the array setting where each and every one of these spots in the array would carry our cross-reactive peptides, allowing us once again to make patterns of which kinases are active and which kinases are inactive in the cancerous cells versus normal cell extracts. And honestly, I think this is just more data with more inhibitors. I think this is probably a nice place to stop. You've got the general overview of what differential sensing is and the general overview of how we apply it to kinase activity. So with that, happy to take questions.